Good afternoon. Am I audible? Okay. There has been a lot of network issues since morning. So I'm just wondering, am I audible? Let me know, am I audible or not?
Yes, sir.
I'm not. Oh, thank you, Adish. All right. Okay. So we will continue with our class on PCR. Okay. Um, If you remember the first class, when you look at conventional PCRs, these are also called as endpoint PCRs because the endpoint is running it on gel, right? We won't know until the PCR reaction is completed whether the PCR was successful or not, right? Only when you run a gel, either you will have, three possible results. One is that maybe you didn't have any amplification at all. Okay. One, the other possibility is that you had amplification and it was perfect, like what you wanted, it came out the way you wanted it. The third possibility is that, you cannot make a decision whether it has worked or not because you were expecting, say, a 1 kilo base pair product, but what you saw was, several other, PCR products also getting formed. Okay, so when that is the case, then these are the three possibilities. When it is not work, then you probably have to go back, check, some of your PCR primers, maybe your your genomic DNA preparations, or your reagents, your polymerase. Most likely, these DNA polymerases are capable of withstanding abuse to a large extent. Unless you have done something grossly wrong, Taq polymerase is a very, very versatile polymerase. It seems to, even when there is a small amount of contamination, it tolerates a small amount of trace contaminants and it still does a PCR for this thing, for a routine purpose fairly well. Okay, but there are applications when things can turn out to be inhibitory. For example, when you're trying to PCR from fossil DNA, right? Now it has become a big fad. So these are called as paleo PCRs. You are taking the human remains, the animal remains from a fossil, and then you're trying to do a PCR, for genomic sequencing or for trying to understand are there any recoverable pieces of DNA. It's highly skilled work to extract DNA from these fossil bones or from some of those old remains. Okay, nevertheless, people have been able to do that. Okay. Um, for example, from the Siberian, those ice, this thing, permafrost, the elephants have, those animals' tissues have been removed. They have been subjected to PCR. Similarly, in the Europe, in the cold mountains and, where they saw some, human remains, right? People who had died and who were, because of the extreme cold, their bodies were preserved very well, and so they've gone back to set up a, isolate genomic DNA, set up PCRs and all that. So all of this has been successful, but it's a very, very highly skilled, work. Okay, because you don't want to be contaminating those samples, paleo samples, with your own sample, because fortunately or unfortunately, we shed a lot of cells, as we prepare. That's why if you see most PCRs, people would put on gloves, people will take extreme precautions not to contaminate with your own DNA, right? So, especially with respect to things like this paleo PCRs or any specialized ones, when people are trying to do genotyping the way we discussed for DNA fingerprinting, they take extreme precautions. Okay, because we know that PCR has this ability to amplify things exponentially. All it needs is a small amount of teeny tiny amount of those contaminating DNA, and your results can become very very messy. Okay. So clearly we saw two kinds of PCRs. One was called as the endpoint PCR. The other thing we saw on real-time PCR, which is like the current, this thing for COVID, backbone for COVID diagnosis. Right? But in between these two, there are all kinds of PCRs. I am not sure if anybody is keeping a tab, but the last time I saw, I mean, this is a while ago, I could figure out about 30 to 35 different types of PCR. Okay. And obviously, each time a person has does a PCR a little differently, they tend to name it, and they give another name for it. So I will quickly show some of these different PCRs, and then, maybe we'll see a short video of that. Just give me a minute. I'm trying to share it. Okay. Hopefully, it will be visible now. Okay. Is the screen visible?
Yes, sir.
Great. So as you can see, there are so many different types of PCR: amplified, fragment, length, polymorphism PCR, allele specific PCR. Where do you PCR, as all of us have one copy from the father and one copy from the mother. Suppose you want to selectively amplify, only one particular allele, then that would be called as allele specific PCR. Then you have ALU PCR. ALU PCR is nothing but ALU is a repetitive element that is found in the human genome. Okay, so people have gone back to quantify the extent of this ALU repeats in the human genome. Okay, simply by doing PCR, you have assembly PCRs, which essentially means you can assemble genomes, short genomes, small viral genomes can be assembled literally step by step on a scaffold like structure. You can do several PCRs, join them together, and then you can have a viral DNA sequence. And that viral DNA sequence people have tried to see if it can be infectious or not. In the case of viruses, it seems to be infectious, but when they try to do that for bacterial species, the bacteria did not come alive. Okay, but we will see. We will see. We will talk about artificial organisms, and hopefully in the end of this module, not in the end of this module, towards the end of this uh, course, we look very short, very briefly, what these artificial organisms are and how they are synthesized. Okay. Then you have colony PCRs. You can have recombinant colonies, and suppose you want to figure out which of them has your gene of interest. You can very quickly do you can screen literally thousands of colonies at one stretch. Okay. Then you have conventional PCR, digital PCR. You have all kinds of PCRs. You can actually see that the list is, okay, there we go. Look at that. That is 37 and counting. Okay, this is what we discussed last time, variable number of tandem repeats, VNTR PCR, where you can actually quantify, how many repeats you have inherited from the either of the parents, and you can use that for quantifying, or DNA fingerprinting, right? So this is what we had discussed in the last time. I will be sharing this by end of the class, so you will have a detailed description of all these things. Okay. These are all, I have purposely kept it short. There were some of them are very big. I've removed it and I have kept it as simple as possible. Okay. So let's look at a short video. Okay. And hopefully we will be able to combine the two, the write up and the video. We will be able to make more meaning out of it. Okay. So let me share that video. Unshare and then share. There seems to be some problem with the video. Maybe. All right. So this one talks mostly about most of those 35 that I showed you. It talks about most of them, not all of them. Okay. But to an extent, I think each of them are variation in the basic theme. Okay.
Several different versions of the PCR.
Is
There are a
the
lot of versions of PCR,
audio
but
audible?
we are going to talk about this red colored one in this particular video, not more about the real-time PCR, because we will be talking about this real-time PCR in a separate PowerPoint,
Is the this thing audible?
Yes, sir, it's audible.
because these are very very important and a little more complicated to understand. So that's why we'll be talking it in separate presentation, but in this presentation we'll be talking about rest of this.
So just a quick this thing, both real-time PCR and reverse transcriptase PCR, both of these are called as RT-PCRs. But whenever somebody says RT-PCR, the depending on the context in which it is being talked about, okay, we could immediately sense whether they are talking about real-time PCR or reverse transcriptase PCR. By and large, these days, real-time PCR has become so pervasive. It is found everywhere, and everybody seems to be using that. People have, when they generally talk RT-PCR, it would be real-time PCR. But there are times when people would be talking about reverse transcriptase PCR. Generally, when you are dealing with RNA as a starting point. So typically for a PCR, you can have either genomic DNA as your starting material for amplifying your gene, which is perfectly fine when you talk about prokaryotes, right? Because prokaryotes, when you look at gene A, gene B, gene C, you may not have any intervening sequences. So all you need to do is take the genomic DNA and set up a PCR. But that is not going to be the case with mammalian cells, or even for that matter, Drosophila or any of those eukaryotes, right? Because they have lots of introns in them. So trying to do a PCR with genomic DNA is going to be a painful thing, right? Why would you, if, for example, one of the gene is supposed to be around 9 kilobase pairs. But if you look at the genomic DNA, right, if you stitch together all the exons, it comes to around 9 kilobase pair. But if you were to look at the the genomic DNA spans literally millions of base pairs, right? Setting up a PCR with millions of 1 million length, a gene which is 1 million base pair long is going to be a failure. You will never get a product which is that big. Okay, you will never get that. Okay, in fact, setting up a long PCR, synthesizing anything upwards of 50 kilobase per itself becomes challenge. So 1 million is going to be too far. Okay, and people don't do that. So obviously, when it comes to mammalian cells or eukaryotes, people will use RNA as the starting material. Okay, they will use reverse transcriptase to reverse transcribe the RNA into DNA, okay, and then they will start the PCR. So that is why this is called as reverse transcriptase PCR. Alright.
Red color thing, which is colony PCR, nested PCR, multiplex PCR, hot start PCR, inverse PCR, long PCR, as well as reverse transcriptase PCR. Now remember, one thing, this reverse transcriptase PCR is also called RT-PCR, R for reverse, T for transcriptase. But again, if you look at real time, it is also R for real, T for time. But RT-PCR denoting a reverse transcriptase PCR, not real time PCR. Because I have found students who confuse this thing. They thought RT-PCR means real time, but actually RT-PCR is reverse transcriptase PCR. So be careful about this part. Rest of the things are pretty clear, and we'll be talking about each of these different types of PCR, but we are not going to talk again about the detailed mechanism of PCR in all these cases. We only touch those lines which are unique to this type of PCRs, and why these PCRs are being generated, or why these modifications are brought about to this PCR. Okay, so let me take a color first. Okay, okay, so let's begin. These are different types, and definitions are given here. I don't think these definitions will help you, so just come here. The first type of PCR that we are going to talk about is colony PCR. Now, colony PCR, as the term suggests, the PCR is done from the extraction of DNA from the colony. A colony means, and we all know, all microbiologists or who have the understanding of microbiology know what are colonies. Colonies means a simple uh, population of bacteria that start to grow on the culture plate or petri plate, onto the food of surface of the bacteria. So you can see these are the colonies. So the colony can be a bacterial colony, it can be a viral colony. If it is a phage virus, so it's giving us plaque. So whatever, it's a plaque or a colony of bacteria, we can take this colony directly from here. So actually colony means it is consisting of different types of bacterial cell, the whole cell, instead of DNA. Now in previous PCR, we have seen we take out the DNA only and then go for PCR. But what happens actually during the research work, if we are researching about, or if we are looking at the genetic expression of a particular type of bacteria, say the bacteria A, for example. Now we need to look for the gene of the bacteria A. We need to purify the gene, extract out the gene, purify the gene, and this is a long procedure before any kind of PCR. So to simplify that, to lower down the time that are consumed, we will start to produce a crude type of PCR, and that PCR is just taking up direct colonies, which are bacterial cells from the plate, and then crack open the cell, take out the crude extract of the DNA, and look for the PCR and amplify that particular DNA with our desired primers. Okay, so this is the basic goal of colony PCR. So suppose we are having these colonies. So what is the procedure that we need to pick the bacterial colony with an autoclave toothpick, and then we put it into the TE buffer. Okay, then TE buffer or TE buffer, and also distilled water. Okay, and autoclave distilled water definitely. Then we need to heat this mixture. So we pick it up, put it into the microcentrifuge tube, and we heat it at 90 to 100 degrees Celsius temperature. It's actually 100 degrees Celsius temperature heating for almost 2 minutes, 2 to 5 minutes heating at 90 to 100 degrees Celsius temperature. After that, we spin the sample. After spinning the sample for 2 minutes at 6000 RPM or so, we take the supernatant. Now the supernatant, so what is the necessity of heating it and again spinning it? Heating this colonies, bacterial colony, will ensure at 90 to 100 degrees Celsius temperature, it will ensure the crack of the cells. So cells will be cracked, the cell membrane will be disrupted, and all the genetic materials, nucleus will be disrupted, genetic material will come out. And sorry, there shouldn't be any nucleus. A very very wrong mistake. Anyways, so cell will be cracked down, then the nuclear material will be coming out to the to the outside, to the water. And then what we'll do, we again after spinning them, what will happen? So in this tube, suppose there are nuclear materials, there are also protein debris and other cellular debris are there. So we need to spin it again, so that in the supernatant or in the top of all these things, we get only the genetic material, which is this, say this DNAs, this red color thing here, and rest of the part is just pellet down at the bottom. So we take out this supernatant, and so this supernatant, now we know that we are ensured that the supernatant is consistent of mostly DNA. So we can provide the primers and take this DNA as the template. So we take out this DNA and put it into another microcentrifuge tube as the template for the primer, template for the PCR. And we know in the PCR, the basics of PCR will remain the same. So we need a primer template, sorry, we need a template strand or host strand, and we also need primer. So we need to add the forward and reverse primer. Then we need to provide them magnesium chloride, calcium magnesium chloride and potassium chloride buffer mixture. Then you need to provide them the DNTPs and also tag polymerase. Then we will set them into thermocycler and program the thermocycler, and the process will begin. So rest of the part remains the same for the normal PCR, but the extra part of colony PCR is that in normal PCR we are taking the DNA, fresh DNA, and going into the PCR. But in case of colony PCR, we are taking out colonies from the plate and then disrupt the colonies, digest the cellular membrane, and take out the genetic material, and then load the genetic material and use that genetic material as a template strand. Okay, for the PCR. So that's why it is different than the normal or conventional PCR. Now let us talk about the hot start PCR. As the name suggested, in hot start means this kind of PCR begins with a very very hot temperature. The temperature like 90 or above 90 degrees Celsius temperature, almost 95 degree to 100 degrees Celsius temperature. So why it is doing in this way? Because this technique is used to reduce the non-specific amplification. Now what happens actually when we first add all these things, all those these primers and templates. The temperature like 90 or above 90 degrees Celsius temperature, almost 95
I
degrees
thought
to
of
100
putting
degrees
full
Celsius
screen, but
temperature.
I think it
So
just
why it is
freezes.
doing in this way? Because this technique is used to reduce the non-specific amplification. Now what happens actually when we first add all those things, all those these primers and templates and all those DNTPs in a normal temperature, say the temperature, the room temperature at 25 to 30 degrees Celsius temperature, what will happen? There is a tendency of the primers to go and anneal with the unwanted region of the DNA or the template DNA. So this is a problem. This is a universal problem throughout the place, that the primer, as the primer region can have sequence similarity with other regions, except for the desired regions, it can go and pair with it. If it gets a lower temperature like the room temperature and so it can go and pair with it. So this kind of wrong or mispairing of primer with the host template or primer with the target template will when the PCR will start to amplify it, so it will start amplifying the non-specific region, it will start amplifying the unwanted region of the DNA. We don't want the expression and amplification of that DNA, but it will start amplifying those regions due to the misincorporation of the primer with the target DNA. To block this, what we need to do, we need to heat the temperature, the initial master mix at 95 degrees Celsius temperature for few minutes before adding the polymerase. This is very very important, right? Before adding polymerase, we heat this master mix to 95 degrees Celsius temperature. So heating this mixture to 95 degrees Celsius temperature ensures that at this 95 degrees Celsius temperature, as the temperature is pretty close to the melting temperature, so it will not uh, make sure that it will not allow the primer to come and bind with the target region. So it will block the non-specific primer binding, so it will block the non-specific amplification of the DNA. Okay, but again for this kind of process we must give them very heat tolerant thermostable polymerase, and again this polymerase are PFU type of polymerase are given in this particular case. Okay, so and this kind of polymers are having a very small activity at the room temperature, but it keeps rising the activity in the higher temperature, right? So this is called the hot start, because at the very beginning we already heat the master mix at 95 degrees Celsius temperature. After that we added our polymerase, and after adding the polymerase again the process begins, melting of the strands at 94 degrees Celsius temperature, then cooling it into 54 or 53 degrees Celsius temperature, then again rising the temperature to 72 degrees Celsius temperature for the polymerization process. So this cycle will be continued, but before all these things happen, what we need to do, we need to we need to add all this, we need to heat this master mix at 95 degrees Celsius temperature, and adding this master mix into 95 degrees Celsius temperature ensures two different things. In each cycle this hot temperature isolates the primer, stopping the molecular interaction. So this is the fact that I have already talked about, that it will isolate the primer out at this high temperature, so primer will not be able to bind with the target DNA in the non-specific site. This is an advantage. Second thing is that hot temperature releases just enough activated DNTP. So you can see here this red colored thing is the thermolabile protecting group. Now what happens when we heat this temperature, this protecting group is just cut out and it will be moved away. As a result of this moving away of this protecting thermolabile protecting group, we can actually carry out this process. Otherwise this thermolabile protecting group actually blocking the polymerization process. This is another important thing about the hot start PCR. We need to make sure that in the master mix we add this kind of modified enzyme which is having the thermolabile protecting group at the terminal. So actually this thermolabile protective group is present in the DNTPs. So the DNTPs that we are providing at this temperature for this hot start PCR must be unique. They must be carrying this kind of thermolabile protecting group at this 3 prime end of the sugar, ribose sugar. Okay, so this is we need to make sure for all. And why it is important, because in normal situation in room temperature the polymerization will not be possible, because this thermolabile protecting group will not allow the polymerase to sit on and adding the nucleotide sequences. So once only it will be heated at 95 degrees Celsius temperature, this labile group will be cut away, and so that the rest of the nucleotide sequences can be added and a polymerization can start. Okay, now let us talk about the nested PCR. Now, in nested PCR again, the advantage and the utilization of this PCR is to lower the probability of non-specific amplification, because this non-specific amplification is a curse to the normal or conventional PCR process. Because in conventional PCR process, the RNA primer can go and bind with different regions, unwanted regions of the DNA. So we can block this unwanted binding of this primer by utilizing the hot start PCR and also by modifying the PCR into the nested type of PCR. Now, in this type of PCR, what we utilize one pair, utilize more than one pair of primers, right? In conventional PCR, we only use one pair of primer, but in case of this type, we utilize more than one pair of primers. Okay. Okay. Now what we'll do here, so suppose we are having two set of primers in this case. Actually, we need two set of primers in this case, which are destined to bind with two different and separate regions in the DNA. First pair is uh, important, and first pair is utilized to amplify a fragment similar to the standard PCR process. So suppose here this is the schematic presentation. So this is the DNA. This is the target region. This is our target DNA to be amplified. So very first primers will be added outside, flanking this target DNA, outside this target DNA region, as you can see here. So it will be added outside the target DNA region. So no primer added inside the target region, outside the target region, and it will start amplifying them for the first time. So it will first amplify this part. So you get the first amplicon up to this region, right? So it will provide and synthesize this part of the segment, and this part of the segment is getting lost now. So we are getting towards the specificity, right? Now the second set of primers are destined to design or designed in such a way that they can pair with the inner region or inner side of this first amplicon, right? So here say the second set of primers will be attached to the inner region. So you can see these are the primers attached to the inner region. Okay, then it will start amplifying the segment so that we get only this part of the segment amplified. This is very very important, right? So you must have two set or two pair of primers. First pair of primer will bind to the flanking region of the target DNA and amplify a segment shorter than the starting DNA sequence. So we are moving towards specificity, and the second set of primers will bind to the inner region of the first amplicon, the inner region of the target DNA, the inner side of the target DNA, and it will start amplifying that inner side segment, and it will start producing our desired product only. It will make sure that we get only most of the part of this specific DNA sequence, but not the other type of DNA sequence, right? In the next slide we are going to see it in detail. We can see here that this is the DNA. So you run the first PCR, denaturation of the DNA, then binding of primers. So primer, this is the first set of primers that are bound in both the directions. Suppose some of the primers may also bind to the unwanted regions, so it will bind unwanted regions. So this is the primer bound to the unwanted region, and these are the primers bound to the actual region, right? Now after that, what will happen? Both of them will start amplifying the segment. So one of the segment produce this product which is totally unwanted. We don't want this kind of product. Now this is a type of problem for the normal kind of PCR, conventional type of PCR. If this kind of primer start to bind with the unwanted region, an unwanted annealing of the primer with the template DNA will result in the unwanted product. So if we get, after all the PCR process is done, inside the PCR tube, if we get lot of unwanted product and very few wanted target sequence, it will be very tedious for us to separate this target sequence out of it. It will take some extra time, it will consume our energy and all these things. So we need to be very sure that from the beginning we get most of our target sequence and least of our unwanted product. So for that, what we'll do now in this process, it is ensured that the very few amount of unwanted product is generated, and we are targeting most of the target sequences. How? Suppose in this product also the unwanted product is generated in this process. So we are getting two different varieties of the amplicon. One is this unwanted, another one is the target. Now what we'll do here, after during the running of second PCR, we utilize another set of primers which you will bind to the exactly to the target sequences. We can see it is bind to the inner side of the target sequences. These two red colored primers bind to the inner side of the target sequences. After binding to the inner side of the target sequence, it will only amplify the segment of target sequences. It will only amplify this part. So what we'll get, we get this part. Okay, so now as a result of that, second type of primers is have least affinity. They are having least affinity towards this part. So no second type of primer will bind with this kind of amplicon. So this unwanted product will stop here. It will no longer move due to the primer specificity that we have chosen. So utilizing two different primers is giving us the advantage of choosing or extracting out exactly our target sequences from the undesired products. So this is the advantage of nested PCR. Now why it is called nested? Because one set of primers are attached to the flanking region of the target sequence, but the second set of primers are exactly attached to the inner side onto the target sequence. That's why it is getting nested in the target sequence. That's why it's called the nested type of PCR. Okay, and again we must before designing the second or primer 2, we must be ensured that this type of primer uh, must have a huge specificity towards the target sequence. So we must know the target sequence so that we can design the primer with great specificity, right? Now let us talk about the multiplex PCR. Now the multiplex PCR is complicated, is totally different than the conventional PCR. In conventional PCR, what we want to do, we just have our target sequences, we amplify this target sequences in huge amount, right? In multiplex PCR, we can run this multiplication of DNA in several different order. That means in normal PCR, we can amplify only one type of DNA sequences. But sometimes scientists need it to, or people need to generate a lot of different varieties of DNA sequences at the same time. we require many DNA sequences at the same time. So how we can do this? So suppose we are having four different strains here, and from these four different strains we need to generate our DNA sequence. And also, in sometimes what we need to know is that inside the cell, we need to know the amount of amplicons that are present and the type of amplicon that are present or not. So for understanding of the presence of any amplicon inside the cell at a particular time, this multiplex PCR is very, very helpful. For examples, the actual application of this multiplex PCR is to carry out many different type of PCR event or PCR event of many different target sequence at the same time simultaneously. So it's a simultaneous amplification of many target interest in one reaction. So it is again lowering our time and also lowering our energy consumption, right? So for example, say these are the four strains and four sets of primers are utilized to amplify four different sets of genes. But again, we need to be very careful about this kind of process, because as we are utilizing four different types of primers, there is a tendency of attaching this primer to one primer to another template and another primer to another template, right? So this cross primer template binding will hamper this process. It can hamper the process, but we need to choose and design these primers with a master efficiency, and also we need to take those strains separately related. So we must not take those kind of DNAs which are similar to each other. If the DNA sequences are very much similar to each other, if we need to amplify them together in the same reaction, it is going to make a horrible mistake, right? So we need to take up the sequences, or the desired sequence of amplification must be of least similarity. Now here, for example, another application of this multiplex PCR is that, as I've told you before, it can help us to understand the presence of a replicon onto a cell or inside a cell at a particular time, and also it is going to give us the idea about the length of the replicon. How, for example, say suppose these two are the different replicons inside, three are the different replicons inside the cell. So how can we know? So we give three different set of primers against the three different replicons. So what we get in the first set of primers bind with the first replicon, second set of primers bind with the second replicon, but third set of primers are free because they haven't got any kind of replicon 3 there. So this replicon 3 is missing, and by utilizing this process we can understand that the replicon 3 is missing, right? How? So this primer 3 is just moving out, it is not bound with this replicon, so it is not amplifying. So after that PCR process is done, amplification of product 1 and product 2 will be conducted. Now utilizing this multiplex PCR process, we can understand, we can tell the length of the replicon as well as the presence of the replicon. So how we can know that? After amplifying the product, what we'll do, we will tag this with several different tagging agent. So normally biotin is utilized during the amplification process. If we utilize biotin DCTP, so we are attaching this biotin to the CTP. So one of the four nucleotides are tagged. So here it is tagged, it is tagging the CTP. So biotin is added to the CTP. So as when the CTP is added during the polymerization process, biotin will be added there. So if we are getting uh, replicon, longer replicon means longer or many more biotin, and smaller replicon means lower range of biotin, and no replicon means no biotin is added. So the tag 1 and tag 2 is added, but tag 3 is nothing to be attached with because there is no replicon 3 is present. Now, this biotin is having the affinity towards the luminex, which is a luminescent dye. Now, this luminex can provide us the insight, and it can give us the quantitative measure of the length of the replicon. Now, as we get here, four biotins are added, so four luminex will be added. So the signal strength that we get from this replicon will be higher, and the signal strength that we get from this second replicon will be little less. So from and then getting the signal and knowing the signal will help us to tell that this one getting as a result it is getting us the higher value, it is getting us the higher signal, stronger signal. That means this replicon 1 is longer, larger than replicon 2, and as a result that tag 3 is not attached with any kind of replicon. Due to the missing of this replicon, there is no signal getting from the third kind of replicon or the tag 3 or the beat 3. So from this one we can tell this replicon 3 is not present. or you can also infer that if the replicancy is present but it is destroyed, so that we can't get any kind of result from it. So by utilizing this multiplex PCR pause process, at the same time after a particular time duration, we can tell the number of replicon that are present, whether they are present or not, and also the length of this replicon. So we can see that if we take these three different samples, three different actually templates to be amplified, and we add forwards and reverse primers, we get, depending upon the expression pattern also, we get different expression levels, right? For example, say this one, sorry, okay, say this one, okay. So say this one is the expression level of this one or the set one or amplicon one is less than, and the second one is more, than third one is much more. So if we get the graph using the capillary electrophoresis, so what we get the results here. So you get the maximum result for this third type of amplicon, and lower smaller result for this type of amplicon, and so on. Okay, so you can utilize multiplex PCR. The advantage is giving us is that we can amplify different desired segment of DNA at the same time in a single reaction. Now let us talk about the inverse PCR. In inverse PCR, the actual thing is that the primers are oriented in the reverse direction of the usual orientation. Okay, and why this PCR is important? And I found this is one of the clever procedure, a very intelligent procedure, very intelligent procedure. How? Let us look at here. It is utilized to clone sequences which is unknown, flanking a known sequence, right? So suppose these are the sequences we need to amplify. This is the unknown sequence. This is also an unknown sequence. So this is a known sequence at the middle, and this known sequence is flanked by two unknown sequences. So the unknown sequence here, and also another unknown sequence here. Now suppose we need to clone this unknown sequence. So before cloning this unknown sequence, we must amplify it via PCR, but we cannot amplify any unknown sequence via the conventional PCR. Why? Because in conventional PCR, we must know the sequence, because knowing the sequence is important to design the primers, right? So if you don't know the sequence, how can I design the primers? So before designing the primer, we must know the sequence, but in this case we don't know this segment of the sequence. It is unknown to us. So how can we amplify the unknown sequence? Now we can amplify the sequence, unknown sequence, but we must have a known sequence right behind it. So suppose in this case, this is the known sequence at the middle. We know the sequence. We can add primers for this known sequence, but you cannot know this unknown sequence. We don't know the unknown sequence, right? So if this is the procedure, that this is suppose this red thing is the unknown segment or unknown sequence, and say get this color, and this part, this rest of the part flanking are the green things are the known sequence. Then we can add primers. So for example, let's say the primer is here in blue in color, so we can add primers then in both the cases, and we can amplify the segment, right? So for amplification of a unknown segment, we must have added the known sequence at both their terminal region, right? But in this case, the total opposite thing is happening. At the middle, we have known, and at the terminal region, we have unknown. So what we need to do, we need to reverse it. We need to put this unknown at the middle and put this known sequence at the terminal. How can we achieve this? This is an actual goal for this particular process. How can we achieve this? We can achieve it by utilizing the light digestion with restriction enzymes. So we make the digestion with restriction enzymes, and after digestion of this unknown sequence with restriction enzymes, a random kind of digestion, it will provide the overhang, right? So it will provide, remember, say this is the segment, it can provide this kind of overhang sequences, and two overhang sequence can make pair with itself. So this is an overhang, this is another overhang that are generated at both the terminal of this DNA, and as a result, these two overhang will be paired with each other. They are joined with each other. This is suppose the joining region. So they are circularized. So they bring close together both the ends, and they make a circularized plasmid-like structure like this. So circular DNA structure is formed. Now what we can do, we know this red colored region or the segment. Now the whole process is joined, the whole DNA segment is circularized and totally covalently attached. Now what we can do, we can use a restriction enzyme to cleave somewhere middle of this known sequence. As the sequence are known, so we can easily provide the restriction enzyme which can make a cut here. So we will provide the enzyme, it will make a cut here. As a result of making a cut here, again this circularized DNA will be linearized, but now the linearized segment will have in the middle unknown region. So unknown in the middle and known at the terminal. Remember, this is our goal. We need to provide this because when we provide this kind of sequence, unknown in the middle but known at the terminal region, we can actually design primers which will destined to attach to the known segment, right? So we know both the terminal regions. So we can design primers which can add, which can be added to this known region and can amplify this unknown segment, right? At the very beginning, what happens actually at the middle was known, at the terminal was unknown. We need to reverse it, and to reverse it, what we usually do, we use light digestion with restriction enzyme both the fragment to provide the overhang, then to make a circularized DNA. Then we use a restriction enzyme to cut it somewhere at the middle of the known sequence to make again a linearized DNA. But in this linearized DNA is completely inverse of the previous type. It is reversed. Now at the middle we are having unknown, which was previously was known. At the terminal we are having known, which was previously unknown. So now this is our goal, to provide unknown at the middle, known at the terminal, so that we can provide the primer which can bind with this known segment at the terminal, so that it can amplify the unknown sequences. So we can amplify unknown sequences by this inverse PCR. Now you can see how much intelligent the technique actually is, right? It's a very, very brilliant technique. Now this kind of inverse PCR can be used to identify the transposable elements, as well as the identification of the genomic inserts, any kind of genomic inserts. Those are unknown inserts. We can detect them utilizing this inverse PCR. Now let us talk about the long PCR. As the term suggests, long PCR, it means it can extend or longer DNA strand using this PCR. What we normally know is that the PCR means the amplification of short target DNA. Usually the target DNA is very, very short, less than 1 kb or like the sequences, right? But if we need to amplify the sequences which are over 5 kilobases, or frequently over 10 kilobases, and most of the time over 5 to 7 or 8 kilobase sequences, which are large sequences. If we need to amplify those large sequences, we can also amplify them via this long PCR. But this PCR is very, very less effective because long cloning or long amplification of the PCR must, for the long amplification, remember, if we consider the polymerization process, DNA polymerase 1 and polymerase 2 cannot accomplish this task easily because they are having a particular processivity. After that, they will fall off. So for that reason, we must need the polymerase 3 during the replication procedure, right? So polymerase 3 is having the beta clamp and the clamp loader protein, with which it can actually hold on to the DNA strand for a longer period of time. So we need that kind of special events or special enzymes in this long PCR process. So we need special mixtures here. We must have polymerase like PFU polymerase, which are specialized, which the processivity of which is higher than the normal type. And also we need the 5 prime to 3 prime, 3 prime to 5 prime exonuclease activity for that. Why? Because as we are amplifying long stretch of DNA, misincorporation is going to occur more, and as misincorporation going to occur more, so we need to delete the misincorporation. For that, we will require the 3 prime to 5 prime exonuclease. In normal type of PCR, in conventional type of PCR, the our interest of stretch of DNA is very short. So as the stretch of desire is short, so the misincorporation of bases is less, and even if the misincorporation happens in small amount, this will not affect the process because it is very, very small stretch of DNA. But in case of this long PCR, as the stretch of DNA is more, so the degree or error will be more. So we need to have this 3 prime to 5 prime exonuclease activity. So suppose this is the template, this is the template strand, and this is the primer. So the primer is added and the polymerization is going on, and one base is mistakenly added. Here C is added against A uh, instead of T. So what will happen? We need to cleave this C out. So we need to have 3' to 5' exon nucleus to cleave this C out of it, and then again incorporate T, which is correct, and then we'll to carry out this particular chain elongation or the polymerization process. Okay, so for cloning of the large genes, we can use it, but it can be only useful if it is accurate. And it is to make it accurate, we need to use the spatial mixtures, and we need to make sure several important parameters. Okay. Okay, guys. Now let us talk about the reverse transcriptase PCR. Remember, this reverse transcriptase PCR is also called RT-PCR. RT-PCR means reverse transcriptase PCR, not the real-time PCR. You must understand this concept. Now, in this reverse transcriptase PCR, as the term suggests, we know that reverse transcription means what we know by about transcription is, suppose we are having the DNA. From the DNA, we are having single-stranded RNA. This process is called transcription. But now there is a modification. If we need to provide
Sir, the video's on mute.
transferred, and it will be transformed into DNA. This DNA is called cDNA or
And
complementary DNA, as we
is it
know.
audible now?
This is called
Yes, sir, it's audible
yes,
now.
the DNA which is provided and
I'll share
produced
the
from
link.
RNA
Okay, so you
using
can
reverse transcriptase
go
will be
back and
called the
revise it.
complementary DNA. Now this complementary DNA, now this RNA strand and DNA strand is there, so it's a RNA DNA hybrid. Now this complementary DNA, this RNA DNA hybrid needs to be dissociated. In the second strand reaction, the digestion of cDNA and RNA hybrid is done using RNase H, and this RNase H will chew off this RNA strands. So we left off with only this DNA strand, and another DNA strands will be synthesized. Now what we get a DNA strand, then the rest of the process will be like the normal conventional PCR. Now this DNA will be separated, the strands will be separated. So here is two strand. Now the primers will be added. So again the primers, for example, say here is the primer, forward and reverse primer will be added. Then the amplification will go on like the conventional type. Now if we look at this process carefully, what we get, this part of the process is a conventional PCR, but for this kind of reverse transcriptase PCR, two steps are more. First stand reaction is a synthesis of complementary DNA from RNA using reverse transcriptase, and the second strand reaction is a digestion of cDNA RNA hybrid, and then providing and producing a double stranded DNA. Okay, after that the rest of the process will be same. Now this
Okay. So few more minutes, but I don't think it's such a mistake. Um, the terminologies have changed, right? I mean, it used to be when people used to say RT-PCR, it generally meant reverse transcriptase PCR. But nowadays, the moment somebody says RT-PCR, as I said in the beginning, it is highly contextual. Definitely, with respect to COVID, it is real-time PCR, right? And depending on the usage, people can mean different things, but you will understand from the context whether they're talking about real time or reverse transcriptase PCR. Okay. So I'll mail the notes, and I will also put the YouTube link. Maybe I'll put the YouTube link in the notes also, so that if there is any this thing doubt, you can go through that. All right. Okay. So we will meet in the next class. Then. Any doubts on this class? Okay. If there are no doubts, then we will meet in tomorrow's class. Okay, we'll talk about the. So this kind of finishes the module on PCRs. Okay. Next tomorrow we'll look at the enzymes that are used in recombinant DNA technology. Okay. We'll probably be looking at things a little differently, but we'll look at it from the application point. All right. Okay. Thank you so much. God bless you.
Thank you, sir.
Yeah.
Thank you.
Thank you, sir.